many things that evolutionary biology cannot explain so far might be explained by host pathogen coevolution, including aging.
So hello and welcome to the Sheekey Science Show, where in this video I spoke with Peter Lidsky, current postdoctoral fellow at the University of California, San Francisco. Lidsky originally started his career studying enteroviruses, and since then he moved to Zurich in Switzerland, where he switched to his focus on Drosophila as a model organism for gene regulation and development. Now he combines his interests in Drosophila and viruses in understanding the evolution of aging. I firstly just wanted to start with your Twitter bio, where you describe your interests as evolution of aging, virology, and Drosophila. And so I just thought maybe you can give some backstory to how you got to the position where you're at today and discuss some of your interests.
Well, there is not much of specific good things to say. Probably since young scientists probably see us, you can put the running title here. All the tricks are made by professionals without a major success. Do not try to repeat at home. So I have been changing my topic several times, and this is not what you should do. But finally I ended up where I am. I think I found a cool story on aging, and I can use this my virology and Drosophila expertise to pursue this puzzle. So I started as a virologist, then accomplished some projects, then decided I will go to something completely different. And then I decided I want to connect both parts of expertise, viruses and Drosophila. And then I found very peculiar phenotypes that are not published yet. And that led to the theory, that aging might be an adaptation, an adaptive suicide, like adaptive death. So, and the aging part so far is mainly theoretical, like math.
Okay, yeah, because in a lot of your more recent work, you describe aging as a paradox. So why exactly is aging considered a paradox?
Well, evolutionary, it's a paradox. The idea of evolution is replication. And if I die by age, I cannot replicate anymore. So it definitely should be bad for my fitness. And if you look, so the archaic view on aging was that just animals in the wild do not survive to the age where they senesce. And that's why aging is sort of neutral. But then, if you look carefully into ecology, we see that a lion of age of 14 has much better chance to die per year than a lion of age of 4. And lions can survive to the age of 14 in the wild. So aging is definitely not neutral. And this is paradoxical, because if lions would live longer, they would leave more progeny.
Exactly. So I think there's clearly several different theories that have been proposed to explain why aging seems to have arisen. And one of them, as you've already sort of described, is this extrinsic mortality, or I think it's the selection shadow. Maybe that's what you're describing. But what are the other current theories of why we age, and why do you think that they have limitations?
Yeah, so the major thing for now is this antagonistic pleiotropy. This is like a pay later hypothesis. So aging is something like college loan. Then we are so strong when we're young, but then we need to pay for it. And if we wouldn't pay in our old ages, we wouldn't be as fit in young years. So it's like a transaction delayed, but it's not clear what is the mechanism, and it's not clear what is the currency. So initially the concept was that there are pleiotropic genes that are good in young years and bad in old years. And if we assume existence of such genes, then this model can work, because still the mortality is there, and much less less animals are surviving to the old ages. So the demographic pyramid is built like that. Here are young animals, here are old animals. Number of old animals is always smaller. So everything that improves fitness in young years should be selected better. But all that requires genes with two opposite functions: good in young years, bad in old years. And how this can work is very difficult to imagine, because no, you know, the unit of mutation is much smaller than a gene. It's a nucleotide. And there is, I haven't found a single convincing example of a gene that kind of evolution cannot resolve. Let's assume related species, and a rat and a squirrel, right? A rat lives 2 years, a squirrel lives 20 years. So rat ages so fast because there are some genes that are missing from squirrel, so this is a completely different mechanism. Or in squirrel evolution somehow managed to uncouple these connections. So these questions are unanswered. So, and if you challenge the people who are like followers of the antagonistic pleiotropy idea, they will either bring you examples that you immediately can destroy with the nature observations, or they refer to experiments. In experiments, sometimes you can see the phenotypes that are sort of predicted from antagonistic pleiotropy, but these observations are not fully investigated, and they can have alternative explanations. And our model can produce these alternative explanations, which are, to me, are more feasible.
Okay, yeah, so from my perhaps naive understanding of antagonistic pleiotropy is that it considers aging itself to be a bad thing to happen, but we can explain it by saying that there's these apparent genes that were beneficial when younger, and as a consequence, aging's like the unfortunate side effects of selecting for these but apparent good genes in the early years, like pre-reproductive age. And so on the flip side, there's also theories that aging is actually adaptive. So what other theories besides the ones that you've come up with seem to suggest that aging itself is actually an adaptive thing to happen?
Yeah, so the adaptive aging theories are also around for a long time, but most of them lack strong. I do not want to upset people who produce them, but I think they are still not very convincing from the point of evolutionary biology, because they require very stringent assumptions. So like they are. There is an idea that the resource release, that we die to give space to our progeny, or this is for acceleration of evolution, or it is for control of population density, that we avoid like overpopulation and collapsing. All this require group selection. And group selection, so I do not want to go in much detail, but it is considered a very infrequent in nature. So like when selection works not on an individual fitness interest, but on the big group, like a group of non-related animals are altruistic to each other, and they are selected as a whole. This is believed to be very rare, because the egoists that is evolving in such an altruistic group can take over all the group with time. Yes.
Okay, I see. So I think that this now nicely leads us on to your theory of aging, or like why aging seems to have arisen. So this pathogen control strategy. So would you be able to elaborate on where this idea came from and what exactly is this theory?
No, so in brief, so we do not use group selection, and this is main advantage of our theory, and what differs us from the previous theory of adaptive aging. So idea is that pathogens, infectious diseases, are the strongest selective factor in nature. So if you look into the evolutionary papers that work with bacteria, so often people use 5% fitness, 10% fitness, so meaning that bacteria which is replicating 5% better than other bacteria is getting benefits, and 5% is by able to eat some substrate that other bacteria cannot. So, and this 5% of fitness gain is something. But if we look in epidemiological model, where let's say we add a phage to this bacterial culture, the phage will kill 99.999% of all bacteria, and the several bacteria which are resistant will get thousands of percent of fitness gain. So, and we do not need to wait while one strain will over-replicate the another. So it is just pathogens are very strong selective force, and we can also see it from genetics. Everything that is associated with immune system evolves much faster. So you can also Google Red Queen hypothesis, to stand in the place. There is a constant coevolution between hosts and pathogens, and to just be in the steady state, hosts still need to evolve its immune system to adapt to evolving pathogens. While we might not see any phenotypic difference, but just pathogens become stronger, host immunity becomes stronger. And even to, like in Alice in Wonderland by Lewis Carroll, to stay in the place, we need to evolve. So that's why we believe that many things that evolutionary biology cannot explain so far might be explained by host pathogen coevolution, including aging. So the basic idea, so if we have a disease which doesn't kill, like HIV, for example, like in monkeys, it doesn't kill much. So, but it sterilizes. So monkeys are less fit, they produce less progeny. Then this infected monkey is rather a problem for its genes than a benefit. And because it doesn't replicate very well, but it can infect its relatives who are located around. And then the genes of this monkey want to kill it. And because there are some uncertainties in pathogen detection, pathogens evolve very fast. They can avoid immune detection of the host. So host might want to kill itself prophylactically, even without information about infection status. And why it wasn't discovered before, because there is a little technical thing I want to add, that because the fact that relatives are interacting more often than non-relatives in viscous population, this fact wasn't very well studied in epidemiological models. So this is like a technical novelty that we introduced, contributed to science.
Okay, yeah, so I think we'll come back to the viscosity element, but firstly, I wanted to talk more about these pathogens themselves. As you mentioned about them sterilizing the individual, is that necessary for this hypothesis to be true, or is it possible that you could also, in theory, pass on the infectious disease agent to offspring, and that could also have negative consequences?
Yes, it should decrease fitness. It can be also that it might kill with the long incubation and infection period. Like if there is a death, highly infectious disease, that is long and lethal in the end also probably can select for shorter lifespan. Now I can tell our last news. So we believe that the Tasmanian devil. I think I told you before, right?
You did, but perhaps we should elaborate to the viewers. But
yeah, so the idea is that there are sort of adaptive death in animals that is called similparity. That's like salmons. The fish goes up the river, lay eggs and die. Very strange type of behavior. And actually in mammals it's not around, almost, with the exception for two clades of marsupial carnivores, Didelphida and Dasaurida. And in these marsupial carnivores, similparity evolves several times, like more than 5 times, independently. It seems to be that there are different mechanisms of this death. And how the mainstream evolutionary theory explains that, it is not clear. Because in all the other mammals, it never happens. Only in this clade, but several times. And we believe that this is a response to very specific diseases that have, so our model predicts that it should evolve in response to diseases with a strong virulence, so they kill, and very fast transmission. They spread very well, because then, so the selection power should be gross to this thing to evolve. And as you might know, in Tasmanian devil, which is also a member of Dasaurida, the biggest, it's like a small dog, like marsupial living in Tasmania. Recently, they acquired the transmissible cancer. These are cancer cells that can go from one host to another by bites, and it's peaking during the breeding season, because during breeding season they fight a lot. And then, if the animal acquired this disease, it will die before the next breeding season. So, and during this time, it still can spread locally, because they also bite around carcasses sometimes. Then they fight for food. But this infected animal doesn't have big replication potential, because it will be probably die anyway. And removing these animals right after the breeding season might be adaptive. We already pitched it yesterday. Our letter went out in Trends Ecology Evolution with this hypothesis. And the curious thing is that this transmissible cancer in devils is now represented by 2 independent lines. One emerged like 30 years ago, and the second emerged like 10 years ago or 15 years ago. And it is specifically curious because there is only 1 or 2 other transmissible cancers known in mammals, so like dog transmissible cancer. And it seemed to be that these marsupials are specifically susceptible for induction of transmissible cancers. And then it would put the puzzle all together. So the if marsupials are susceptible for induction of transmissible cancers, then they have metastable epidemics. They induce it. They have these epidemics of virulent transmissible disease that kills most of the animals, like it happened in Tasmanian devil, and disease goes away or adapts, becomes more benign, as we predict from the evolutionary parasitology. And then a new cancer is induced again, and again the thing repeats, and this can put a constant selection pressure to evolve similparity. So this is like still a hypothesis, We are working on modeling all that, but at least it is something that can explain. It can produce a better explanation than mainstream model.
Yeah, no, it's a really interesting example. And as you mentioned, this is only relatively recent, these clades. And so, I mean, clearly they were the ancestor before these clades separated also was aging. So I guess from my understanding, you're saying that now we have this additional observation of this transmissible cancer. This might further select for like increased similarity, or maybe I just misunderstood.
Yeah, the devils now are not simiparos, and maybe in devils, similarity might not evolve because of the population structure. So that's what we are now studying. But they just are exposed to these cancers. They do huge population ups and downs because of this. And others that evolve similarity, they can control cancers with just killing all the males, or in some species, both genders are dying after the mating season.
Okay, I see. So this is kind of like a way that you can actually test the model that you proposed in
Sort of. So the prediction from here is to find transmissible cancers in other marsupials.
Okay, yeah. So besides looking at the transmissible cancers, in terms of like other lines of evidence that supports your theory, you've mentioned already how like viruses, etc., have a strong selection pressure because of like the red queen hypothesis. But what is the strongest evidence to actually support this hypothesis that you're presenting?
What is the strongest evidence? Well, I think it's a matter of taste. So we have classified our all the observations, and we think that our model can produce a better stitch to the reality than the classical model. So, for example, birds and bats, everything what flies, lives longer than terrestrial forms. And it is not about being bird, because the non-flying ratid birds, they don't have extremely long lifespan for their mass. So it's about flight, not about something else. Why do the typically people say, okay, this is because they are better protected because they fly and the cat cannot catch this sparrow. But if you look in ecologic data and look for annual mortality of sparrows, you find it's very, very high. So it's like 60-70% per year. And a sparrow can live 20 years, which is completely kind of off for a terrestrial animal. And this is not clear why. So the correlation between longevity and flight doesn't have explanation in classic theory. So, but we propose that it is because of this viscosity. So for our model, it's necessary that relatives are interacting more than non-relatives to infect them. And if the population is well mixed, so that interactions are more promiscuous, then long life should evolve. And it is known that dispersal in birds is like about 10 times bigger than in terrestrial animals of their sizes. Yeah, I probably need to specify that this interaction between relatives is needed to run kin selection. So because if I will kill myself to save everyone in the audience, my genes will not proper from that. So I need to sacrifice to help my own progeny, and only that will promote the evolution of my genes.
Okay, so like to sort of summarize, flight organisms that can fly have like lower viscosity, so they're mixing about a bit more, and so there's therefore a weaker selection for aging because they're less likely to pass on any infectious agents.
Yeah.
Okay.
Yeah.
So then what does that tell us about so that's like flight versus terrestrial, but what about I guess like early organisms where like more sea-based. Water, like aquatic organisms versus terrestrial. If we think back to like when did aging first arise, or like could it have arisen multiple times? What can we learn about, like using your model? What can we learn about when did aging first like evolve adaptively? If that makes sense.
No, well, I'm not a big expert in marine life forms, but you probably moved the discussion towards organisms that do not age, right? Like hydras or planaria. Is it your intention?
We can head that direction, sure. Yeah, that wasn't actually my plan, but it works.
We wrote in our review that they probably cannot mechanistically discriminate between reproduction and regeneration. And if you cannot disconnect these two things, then you have to stand the high infection burden. On the other hand, also hydras, which are non-aging, they can switch to the aging form. And I'm not a huge expert, but I think they do it in response to stress. And it might be that just if they are infected, they rely on the pathogen detection and switch to an aging form to clear the trouble, and then they go to the non-aging again. So, this is something that I need to read more about.
Okay, yeah.
So generally what swims in the ocean behaves a little bit different, because pathogens can also diffuse across bigger distances. Then also vertical transmission is low, I believe, is lower in the life forms that are not viviparous. So the rules there are a little bit different.
Okay, yeah, I see. The other thing that made me think of though are like plants, and obviously they're fixed. I mean, they obviously move, but they can't. They're not like animals that move about, and so there are many like very long-lived species of trees. Like how does they also get infected by different agents? Does that model of aging in plant-based organisms fit in with your model as well?
Yeah, we were thinking very superficially about that. So one need to model that accurately, and I'm afraid I cannot answer that. In principle, yes, it can be true. So why trees are living so long? Of course, to grow large, you must live long. So to use the benefits of the size, you need time, and this benefit of the size can be bigger than the penalty posited by pathogens. Right, so there is always trade-off. So the virus pathogens are also around in plants. They are transmitted by insects. In principle, it also can work. If you think about plants that drop leaves and evergreen plants, then you might find that evergreen plants typically have some better protection against insects than others. But I'm not sure this is a strong argument so far.
Okay, yeah, that's still somewhat compelling, I would say, though. But I think one of the things that I find interesting is just firstly considering that aging could be adaptive in this situation, and then what it tells us about like how many times aging might have evolved, whether there was one like universal ancestor that got infected, and like whether there was like co-evolution of the origin of like virus or whatever this first infectious agent was and aging itself, or whether you've even like thought that far back.
No, so we believe aging is part of the immune system. It's not a virus thing. So it's a host protection that benefits the kin and not the individual itself. just to make sure we're on the same page. So the answer to your question is I don't know. So I think what we see in nature tells us that aging is quite conserved. So I think it's reasonable to hypothesize that it was the single event. But the problem is that still aging is not defined. And we know that something bad happens with age, but the clear scientific definition is still missing. And with this this input, it's very difficult to speak about evolution. It seems to be that conserved genes are involved. That's pretty much it.
Okay, yeah, so kind of like on from that, obviously there's been a wealth of studies that have investigated like different like aspects of aging, senescent cells, proteostasis, autophagy, nutrient sensing. Like, how does this theory fit into the current observations made in the so-called different hallmarks of aging?
Which observations are you speaking about? So
any of them. So like, how does the observations that we've observed to extend lifespan in different organisms in the lab fit in with this hypothesis?
I don't think that there is some contradiction to whatever experimental data we observed. So the problem with hallmarks of aging is that it is not clear what is the reason and what is consequence, right? So if so the general again, coming back that aging is not defined, it is a problem, because if we don't know what it is, we cannot figure out what is important, right? So if we like people are getting myopic with age. This is a favorite Vadim Gladyshev argument. And so if we prescribe them glasses, we can increase their because myopic people are hit by cars, let's say. So if we prescribe glasses, then we increase survival, we increase life quality, but we don't cure aging. And same can be true on biochemical level. We targeted mitochondria, get a little bit better result, but it might be secondary effect. And the problem with these hallmarks is that it is not a structured like sequence of events. It's a number of observations. They do not contradict our model anyhow.
Okay, so how do you then think that having this model in place will help us, like as researchers, better design experiments to find out what aging is?
No, so we believe that, as I told, that aging is part of the immune system, and then it gives some handles, because some approaches gives predictions. For example, it's known that the chronic infection can accelerate aging. People who live with HIV, even if they're on therapies, they are on average 5 years older according to methylation markers. So this is what is predicted by our model. So we believe that immune system is something that should orchestrate the whole process. And generally the idea that aging is adaptive is a big deal, because let's say as an example, these parabiosis experiments, right? So the two mice, one old mice, one young mice, got connected. The old mice gets younger, the young mice gets older. So it has been done like 20 years ago. And for a long time, people were trying to identify factors from young blood that can make old mice younger. But like in 2019, I believe, Conboy's lab, they published that just replacing plasma with albumin
Yeah.
works almost as good as young plasma. So it's not meaning that aging is not a loss of function, not something is missing from the old organism. It's gain of function, something emerges in the old organism. And I think if the idea that aging can be adaptive would come before, then it would be less time between these two experiments than 15 years. So, but this focus on aging as a stochastic, entropic, generally detrimental effect moved focus to, I think, in the wrong direction.
Okay, so obviously the immune system is incredibly complex, to even in humans, but probably many species. But how exactly do you think that we can now use this more focus on the immune system to understand ways to actually intervene in the aging process? I mean, you just mentioned the example of blood dilution, but what exactly does that tell us about how to target the immune system, given that obviously it has benefits in terms of preventing us from diseases?
Yeah. So good question. So we are working on that, and we are in a not in a advanced stage of it, but we also believe that cellular senescence is a immune thing by evolutionary design. So because, as you might know, cellular senescence has been proposed to be anti-cancer thing, that if the cell undergoes neoplastic transformation, it activates a pathway and gets into this senescent state, and then it's cleared by immune system. The question is, why so complex? Why don't go into apoptosis? But then, when people studied in more detail, they found that presence of these senescent cells is very cancer promoting. So everything I think initial transformation and angiogenesis, metastasis, proliferation, everything goes worse for a patient and better for cancer. And it is not clear what for senescent cells are needed. And we believe this is an antiviral story, that the infected cell somehow activates the pathway and signal the immune system. And then this antiviral story became involved in adaptive dying. And we believe that virus, if it is antiviral, then viruses must have interactors that can affect the affect senescence. And that's what we are doing now. Because of my virology background, we are trying to find interactions between viruses and senescence. But also, very importantly, our model proposes a huge support for senolytic therapies. Because, as you might also know, many evolutionary biologists think that if senescent cells are there, they are good for something, and removing them might be either detrimental or not very helpful. So our model suggests that they are here just to kill us, and senolytics must work.
Mm-hmm. Interesting, because I think I remember you mentioned senescence when I met you almost a month ago now at the other conference, and I believe you also have done some preliminary work with p53, which is obviously kind of my interest, but important in terms of understanding this decision between cell death and entering senescence. So, if you're willing to, would you be able to remind me what it was you've done so far with looking at P53 in terms of this response?
No, well, I think for broader audiences might be a little bit premature because we don't have mechanistic data so far. We just have phenotypes. But I want to share that, P53 is a key protein for cellular senescence. It has been discovered, correct me if I'm wrong, 6 times by 6 independent labs, and 5 of them were virologists. So because it was found as an interactor with the big T antigen of SV40, and some groups identified it first as a viral protein because it accumulates in the nuclei of the infected cells. So big T antigen is inactivating P53. So for sure there is some interaction. So what we see in Drosophila, we see that it seems to be that P53 somehow is involved in detection of the chronic infection and shortening the lifespan in response to chronic infection. But these results should be still considered as preliminary.
Of course, yeah. But definitely super interesting. And I mean, there's obviously still issue is not the right word, but obviously in terms of defining what a senescent cell is something that's like with aging, being able to define what is aging within the senescence field, it's still not clear what exactly is a senescent cell given the sort of heterogeneity. But I suppose it also links in with the fact that P53 itself isn't universally found in all organisms. Maybe there's like proteins that are very similar to it, but it does suggest that, again, that there's an element of universality to this mechanism, but in different species there's going to be slightly different mechanisms at play.
Yes, I believe that P53 is DNA damage response, first of all, right? And correct me if I'm wrong, I think in worms we don't have much of cellular senescence so far, right? So cellular senescence, I don't think this is a universal universal mechanism. So but I think the primary role of P53 is response to DNA damage.
Okay, yeah, so that's kind of exactly my point is that senescence might explain the aging process in humans, but for other organisms where we see aging, that still fits your hypothesis, but the mechanism's going to be different.
Yeah, mechanisms can be different because the hypothesis proposes the adaptive death is good in epidemiological models, but the mechanism of this death can be very different. So like in this case of similparity, salmon, it's not aging at all. It's like cortisol shock and a response to it. So there can be a huge variation of ways to die.
So, besides what you're currently working on, like, what are the other sort of potential limitations of your own model to be like your own sort of critic?
No, well, so it is still much better than anything else. So, but as you might know, in science, we are never in the end point. So we are approaching the truth as we think, but we never are we never got to it finally. So there there are I asked many people to find holes in my model because so I want to know it first. So what they found is like that we can model very well how in population of short-lived animals, how if the long-lived mutant emerges, how it get destroyed, eliminated, how the limited lifespan can be preserved. But what if evolving short life starting from a long-lived population? So this we didn't model very accurately. I think we still can, but on the other hand, there is a thing called Cope's rule that at least in mammals, evolution typically goes from smaller life forms to bigger life forms. So the small animals evolve big and then get extinct. And since lifespan is correlating with the size, so it's expected that it always increasing in evolution, and evolution back is not a very frequent event. No, so this is a small small disadvantage. Yeah, no, of course there are like quantitative arguments. Okay, you say pathogens impose a huge selection pressure, and that can explain aging. But can you measure this selection pressure? So we cannot yet. So, but the same arguments can be added to any other model of aging. Like if you say pleiotropic genes evolution cannot disentangle beneficial and detrimental traits, how strong this connection is.
Yeah, that's a good point. So, like, the thing about evolution that I think, I mean, at least I tend to forget about it, is the fact that it's still happening today. Like, it's a continuous process. Like, even as the human species, we're still somewhat, well, evolving, albeit maybe different rates to before. But given the model that you've now got in place, how do you, like, if you could predict, like, hundreds years or, like, looking into the future, how do you think that our like role in the planets is going to influence the rate of aging in other species going forward?
No, so hundreds of years is nothing, of course. Hundreds of millions is something. So, but for humans, I believe that we are evolving towards extension of the lifespan, because we already resolved most of the pathogen problems just with the sanitation and now vaccination and drugs. So I think in a couple of millions of years we will get much longer lifespans.
Awesome. And so to kind of go back to a point that you made at the start, which is that you've obviously been exposed to many different areas, not just of biology, but also maths and your previous research before now. I was just curious to know, because I always like to ask this question, like what sort of interesting books have you read, or things that really sort of helped you to sort of create this interesting thought process? Or so basically just any sort of book recommendations.
The books?
Yes.
So, no, I think I will be not very original. I must be, but I can't. So no, I read this like Watson, the like molecular biology of gene, very very dry, and the Dawkins.
Like the selfish gene, I guess. Or...
Yes, selfish gene. There are still, I think still the problem of sex is not resolved. He is claiming he did it, but I don't think it is resolved, and it also might be connected to infections.
Okay. So there's also a lot of excitement in like the longevity aging space at the moment, in general, and there's like things like cellular reprogramming that seem quite exciting. I mean, are there any things besides focusing on the immune system that you think is going to be quite interesting in the next few years?
No, well, things are changing so fast. No, generally there is a lot of works going on. I'm a little bit concerned that, like as a virologist coming to a different field, I'm a little bit surprised that people consider the observational definitions sufficient. It's like if we would like to cure COVID, and we would say we see that there is high temperature, we see cough, we see vomit, we see whatever, and we want to classify all these symptoms and invent a pill against each of them, and then we will cure COVID. And we don't need to study virus because who cares about virus? Important thing is damage that the virus does to the human body. So I think in case of viruses, it definitely didn't fly. So in aging field, instead of microbiology or virology, we have evolutionary biology. And it is still somehow, I think, underscored. So people think that with the big data set, multi-omics, they will map every protein in the human body and understand everything. So it still might, still like the Monte Carlo method of looking for things, like searching a little bit here, a little bit here, it might get us to some therapy, but I think chances are low. So I think we historically science worked like we ask simple questions, we answer simple questions, we ask complex questions. Also in medicine, you first get to diagnosis, then you think about treatment. And here I think this principle is somehow eroded. Nevertheless, even with the without a solid trajectory, we can get to nice treatments. I'm not sure they will be final solutions, but they might help.
Well, yeah, I'm glad I've been able to give you this platform to speak about your hypothesis because, I mean, I think I was lucky that, at least in my own degree, I had some sort of lecture series on evolutionary biology. I would have to say that it was my weaker point compared to biochemistry, but I agree that it's definitely important to always be aware of the the bigger picture as opposed to just hyper-focusing on what genes are for, as you say, the observational approach. So do you have any other words of wisdom or words of advice for current students or just anyone listening that you would like to share before we wrap up?
My career is not the best example to trying to repeat, so probably this might be not very helpful. So the doctor, cure yourself first. But and I think now academia is super competitive, and you need to think about every step. You don't want to make I will make this decision and a couple years later I will look what happens. So you need to plan very in advance and chat to people around, specifically when you choose a lab. You need to first thing you need to do is to look what happened with people who left this lab recently. Of course, it's a super exciting time to be in science, but it's a very hard time to be a scientist now. But of course, excitement overweight all the other things.
That's true. Well, I'm glad that we've ended on a high point. It's been great talking to you, and for my viewers who want to follow your research, I can put your Twitter in the description, but is there anywhere else where I can send my viewers?
Okay, good. Thank you. Sorry, I was speaking so slowly today. I think and I think my accent got even worse. Seemingly impossible, but it got. Anyway. Okay.
Well, thank you again for coming on. It was a really interesting and thought-provoking discussion. So I'm looking forward to see some of your more of your findings as you are able to sort of provide more evidence to support this hypothesis.
Yes, working on this 24/7.
Exactly. Nice.
Okay, ciao.
So I hope you've learned something in this video. Thank you to my Patreon supporters and thank you for listening.